It is my great honor and pleasure to introduce to you today today's speaker, my good friend, Dr. Jenny Gumm. Just wanted to give you a little bit of an idea of her background, because I think it will be of interest to a lot of people here. So she got her PhD at Lehigh University. She got a postdoc at the University of Maryland, Baltimore County, which is where we first met. And then she was an assistant professor at Stephen F. Austin State University before taking her current job as a supervisory fish biologist, project leader, and facility manager at the Ash Meadows Fish Conservation Facility for the U.S. Fish and Wildlife Service. And her research has really focused on a number of animals, but really on fish, behavioral ecology and sensory ecology of fish. She's worked in mollies, cichlids, damselfish, darters, and then now she is in a conservation position and is sort of responsible for the conservation of the devil's hole pupfish, which, if you aren't aware of these fish, you're going to be hearing about them today, and they're pretty amazing. And conservation-wise, it's a really interesting species. So, with that, Jenny, take it away.
All right. Well, hopefully our internet issues are sorted out now. But thank you for the introduction. I'm excited to talk to you today and tell you a little bit about what I do now at the Ash Meadows Fish Conservation Facility. As Megan just told you, I had a pretty sort of standard academic path for the majority of my career, and then had a pretty major pivot when I left my academic position to take this position. But I had worked on pupfishes for a really long time, focusing on sexual selection and reproductive behavior. And so it's sort of just my background and experience had sort of put me in this place where I had a little bit of expertise, and being aware and working on pupfishes for so long in my career, everybody knows devil's hole pupfish if you work on pupfishes, and they're actually surprisingly well known by a lot of biologists because they are often used as examples in evolutionary biology textbooks for small populations, conservation biology textbooks for the history, and all sorts of other interesting things. So I'm going to tell you today a little bit about the history and conservation efforts for the species, and then what we do at the Ash Meadows Fish Conservation Facility. And I have no embarrassing pictures of Megan in my talk. Just in case you were wondering, you can relax a little bit. Please, if you have any questions, feel free to reach out on Twitter, or my email address is on the bottom of my slide here, and then at the end as well. Especially if you have questions about, if any students have questions about jobs or anything like that. There are going to be a lot of internship fellowship positions coming up soon for the Fish and Wildlife Service, and we are very much interested in trying to diversify our service and our workforce in conservation biology. So we try to preserve biodiversity, and we think diverse ideas and diverse people are the best way to do that. And so if you are interested in joining Fish and Wildlife Service ever, feel free to reach out, and I'm happy to tell you what I know. So the first thing that I want to say about working with the devil's hole pupfish and working in conservation in general is that it really relies on partnerships. So I'm with Fish and Wildlife Service, and I'm actually in the refuges program, but we also work very closely with the Fisheries and Aquatic Conservation Program, as well as the Ecological Services Program. And we have partners in National Park Service and the Nevada Department of Wildlife as well. So the three agencies collectively manage the species and the habitat where they live. And we have about 25 regular collaborators. And we were doing conference calls before they were cool. And we continue to have, well, everybody has like 4 or more conference calls a day now, or Zoom meetings, but this is sort of standard of how this has been managed and how the coordination has occurred for a really long time for this species. So every morning at 8 a.m., all of these people from all of these agencies get together and talk about what happened each week with the species. So teamwork, working together, group work for students out there, all of those things are really critical to successful conservation initiatives. So this is a really tiny fish, and today I'm really going to focus on the devil's hole pupfish story. Um, and while they're only about an inch big, they are a conservation icon. They occur on the Nuuvi and Nuwu ancestral lands in the Ash Meadows area. It's an area that is a natural oasis. There are about 50 plus springs and seeps that occur that it also in that area, they have about 26 endemics, which is some of the highest endemism in North America. So it's a really special area, and it's been used by a lot of different tribes as sort of a meeting group or a meeting area in the northern Mojave Desert. It was first described by settlers in 1849 that were going out for the gold rush. And Devil's Hole itself, the habitat, was known as the miner's bathtub at that time. Devil's Hole pupfish was described in 1930 by Wales, and it was first protected in 1952 by President Truman as part of the Death Valley National Monument. It was formally protected as a species, not just because of where it lived, in 1967 as part of the Endangered Species Preservation Act. It was one of the first group of species that were preserved legally in the country. And then this act was formalized more into what we know now as the Endangered Species Act. The other reason that they really are historically important to conservation biology is that in 1976, there was a landmark ruling by the Supreme Court, the Capert versus the US case. And this was a fight over water rights. There was pumping going on in the area, and the water in the habitat had dropped. There was an emergency injunction put in place, and that went all the way to the Supreme Court. In reading some things about this recently, I found it very interesting that it was a unanimous decision in favor of the fish and in favor of water rights for the fish. Because I think in this day and age, thinking about the Supreme Court making unanimous decisions is probably pretty rare anymore. I can't think of the last time I heard of that happening. So it was protected, and this was the first test of the Antiquities Act, I believe. So it remains important because of that historical role that it played. So Devil's Hole itself so I'm in Las Vegas right now, and Ash Meadows National Wildlife Refuge is about 90 miles northwest of Las Vegas. And on the grounds of the National Wildlife Refuge, there's a 40-acre plot that is actually part of Death Valley National Park. So it is on national park land, and you can see that on this Google Earth image in the middle here, it's a hole in the ground. It doesn't look like much, and if you're driving by, there's a good chance you wouldn't notice it because it's just a fissure in some of the rocky areas. So this is on the bottom left, a historic picture of a platform sort of going over the hole and the fence, the old fence. On the bottom right, you can see what it looks like now. It continues to have a platform. It is open to the public. You can go and look down at the water, but there is this cage over that platform because people tend to throw stuff in the water. A different view from the water's edge here, looking up, you can see that platform and fenced area. And then on the right here, you can see a little bit closer view looking down at the water. For scope, there is a diver back here in the water. And so you can see it's a pretty small habitat. So if you're looking at the picture on the right, this platform we deploy every time that we need to work out in the habitat, but usually it's not deployed. That is about 20 feet by about 12 feet. And the area under that platform is about 18 inches deep. And then the area beyond the platform where you see the diver actually goes much deeper, and it's a cave-like environment. So this is a very extreme environment, and the Devil's Hole pupfish is the apex predator in the habitat here. Not many things can withstand the conditions of Devil's Hole. So it is geothermally heated to about 33.5, 34 degrees Celsius. And there is very little dissolved oxygen. I think it's about 2.5 milligrams per liter. And those are really the main extreme variables in terms of the environment, but it's also a very energy limited system. So you can see in the picture on the left that the sunshine is hitting the wall here and sort of hitting the water's surface, but this only occurs during part of the year. There's 4 months of the year when no sunshine hits the water's surface at all. So there's no primary productivity and pretty much nothing driving the energy web or energy flow and food web through the system. So that energy limitation is another component that limits what can and does live in the habitat. I mentioned the deep area, and you saw the scuba diver in the last picture. This is a cave habitat. Pupfish live down to about 80 feet or so. Divers have been down to about 450 feet with a sight line beyond about 520, and beyond that, we don't really know how deep Devil's Hole is or necessarily what it connects to. You can find, if you Google Devil's Hole earthquake, you'll find some really cool videos of seismic seiches occurring on the surface. The earthquakes that we had in the summer of 2019, the water sloshed all the way up above this game camera over here on the right-hand side. And it was about a 15-foot wave action going back and forth. So the habitat is influenced by earthquakes, not only ones that occur near here, but as far away as Alaska and Indonesia. We've seen wave action in the habitat. So that gives you a little bit of context for the species in general. The biggest thing about the Devil's Hole pupfish is that it does have a very small population size. They have been doing counts of the population the same way since 1972 and estimating abundance through scuba counts. So we do think this is a good estimate of total abundance of the population, and this is the only population of the species. So what you're seeing here on the top line, the sort of grayish blue, is the fall count, and then the green line on the bottom is the spring count. And for a pretty long time, there was some good variation between fall and spring. This is thought to be natural because it is from November to February where you have no energy input to the system. So the population tends to get smaller during that time. And historic population sizes in the fall were somewhere in the 400s or so, and in the spring around 200. There were some major population crashes in the late 90s, and then again in the early 2000s. In 2006, and then again in 2013, the population was less than 40. The scuba counts found 35 and 38 fish total on those dive counts. So it was at these two time points that some critical management decisions were made. And those both relate to the facility that I'm going to tell you about today. In 2006, they basically said, let's make a facility and started to get funding for it. And then by 2013, it was built and they rushed it into commission so that we could receive eggs from the population. So since 2013, things have been looking up. In 2018, that we had the highest count in about 15 years. That was 187 fish in the fall. That was really cool. That was like 3 weeks after I started with Fish and Wildlife Service. So that was a fun day to be there for that count. But in general, we do see an upward trend right now in the population. We don't know anything about 2020 because all of the diving has been shut down due to COVID limitations. So we hope to get back into the water in January of next year. So regardless of sort of the historic numbers, in the last 15, 20 years, the population has not been above 200 in the wild. And that's a really interesting place to work psychologically because we're facing extinction on a daily basis. a population viability analysis that was done in 2014. This is showing probability of extinction in some time frame. And so it's a little bit different depending on the fall and the spring counts, because the variation in those counts is a bit different. But in general, the mean time to extinction is about 30 years, and median is less than that. It's about 26 or 27. So the efforts to recover the population or conserve the population are done sort of in this framework of knowing that there is a high probability that the species is on its way to extinction, which makes for a really interesting sort of context to do our work in. Another way to look at that is looking at the probability of extinction over time. If you look at it cumulatively, either based on fall or spring numbers, there is an 80% chance that the species will be extinct in 50 years. So this definitely gives us a context for our work and sort of flavors some of the things that we do in terms of making decisions and taking actions for the species. So the recovery plan itself, despite this sort of short time frame for thinking about extinction, the recovery plan for the species has not been updated in almost 30 years itself. So we've got a recovery plan from 1990 that has not been updated. As many recovery plans do, our recovery plan does have a refuge population and captive breeding program as part of the recovery actions for the species. These captive breeding programs in general tend to supplement wild populations. They can also establish extirpated populations, or they can just be backups. We call these lifeboat or ARC populations. And this is where ours falls. The population that we maintain at the fish facility is just a backup in case anything happens or in case the wild population does go extinct. So some well-known examples that you might be more familiar with that have had refuge populations and captive breeding as an important part of their recovery include California condors, which at some point all of the wild condors were taken into captivity. A breeding program was established, and then now they've been repopulated onto the landscape. And then black-footed ferrets have a similar sort of story. All refuge populations and captive breeding programs have challenges. Depending on those goals, you can have challenges with the population just becoming self-sustaining. Reintroductions are always tricky. The population that you have in captivity itself needs to be genetically diverse. There are issues with domestication and disease oftentimes in captive populations. And then there's just the administrative challenges as well. There are always financial and physical resources, especially when you have both wild and captive populations. People come at conservation with different philosophies in terms of where money should be spent. And there's trade-offs in different types of approaches. There's also administrative continuity, having people that know the history of a program and have that institutional knowledge of being able to work on some of these systems is really important. So having refuge populations for the devil's hole pupfish has been an important component pretty much since they were preserved or started to be of conservation interest in the late 60s. There's been 3 refuge populations. They are just 100 or not, they're about 1,000 gallons in a concrete tank. And all of these failed. So 2 of them were lost to mechanical failures. So pumps just went out, and then temperature fluctuations caused the populations to be lost. And then the third one, this Point of Rocks population, which is also on the Ash Meadows National Wildlife Refuge, but it's located very close to another spring where there's a different species of pupfish. And a few of those found their way into the refuge population. So the population of devil's hole pupfish was considered lost to hybridization. It's because of these failures that in 2016, when they started thinking about another population or another refuge facility, they thought about doing things a little bit different. And so the Ash Meadows Fish Conservation Facility really is a grand experiment, and there's nothing like it at least in Fish and Wildlife Service, anywhere else. So this facility was built solely for Devil's Hole pupfish. It doesn't look like much. It looks mainly like a little garage. Devil's Hole is about a mile away, just up the hill. But inside this garage is something very special, and that is a replica of Devil's Hole. So this is to show you our population right now. I don't know. We'll see how video over video goes. This was just a couple of weeks ago to show you our happy, healthy, thriving backup population of Devil's Hole pupfish. They look great. We've got some of our monitoring equipment there, hobo loggers and some invertebrate collectors. But this is, while the hole in the garage doesn't look like much, it does house this really special population. So, oh, there we go. So this refuge tank was built a little bit differently than those previous attempts. This is actually 100,000 gallons of water, and it was built to mimic Devil's Hole as closely as possible. So we have this shallow shelf area out in Devil's Hole. We have a similar shallow shelf area in the refuge tank. Really scanned and carved out of styrofoam to match exactly the shallow shelf of Devil's Hole. We've got the deep pool in Devil's Hole, and we have a deep pool in our refuge tank as well. Obviously, digging a hole 500 feet deep was not possible, so it's only 22 feet deep, but most of the water volume is in a cavern-like environment. So you still get that cave experience if you're a Devil's Hole pupfish in the fish facility here. We do also keep the parameters a lot closer to Devil's Hole in terms of the environmental parameters than those previous attempts or refuge populations. So we keep it a little bit cooler, and we keep the dissolved oxygen a little bit higher, because previous work has shown that this will increase reproductive output. So as we're trying to get this population self-sustaining and viable, those parameters are a little bit nicer in terms of stress to the fish. But we have control over these, so we can adjust the temperature as needed. There's some differences in the community ecology. The wild population is crustacean abundant, and ours is more insect dominated. We also have some differences because we don't get that earthquake effect. So we get buildup of algae and detritus on our shallow shelf that we have to manually remove, as opposed to out in the wild where a nice earthquake comes in or a flood and cleans it all out naturally. A number of studies have been done looking not only at those physical measurements, which I just highlighted some differences in temperature and dissolved oxygen, but we do have some differences in nutrients as well. We've got some different nitrogen types, and the nitrogen to phosphorus ratio is a little bit different at this point. And if we think about this as an ecosystem, it really is supposed to be a replica of the Devil's Hole ecosystem. And so we can think about how this is going to change over time. And some of these things are expected as you would think of succession in a natural environment. We know that there are differences in the biotic features as well of the community. This study also looked at the microbial community, and I'm not going to go too much into this, but the top lines are Devil's Hole samples. The bottom bars are samples from the fish conservation facility, and the different colors are different groups of microbial organisms. And so we have a lot of cyanobacteria out in the wild population, and we don't have much cyanobacteria in the fish facility at this point in time. And then we have some other species that or groups that are highly found with quite a bit more frequency in our captive population than the wild. So this, again, these samples were taken very soon after fish were added to the refuge tank. And so this is something else that we need to revisit and see how it's changed over time. I also mentioned the invertebrate community. One of the main differences that we found that we have to actively manage on a regular basis is some predaceous diving beetles. We have 3 species of predaceous diving beetles that have been found in the refuge tank. And these are problematic because we also witnessed them attacking and consuming larval pupfish. So that's not great. This is our research tech, Mitch, and he manages the beetle population. It's always important to celebrate your successes. So he got a cake when he had reached 10,000 beetles captured in the refuge tank. And this is something that we do on a weekly basis. Right now we're collecting twice a week to try to manage this predation pressure. And if I updated his cake, it would now read 25,000 plus beetles collected from the refuge tank. But this is going to be an important component to the rest of the story as I move along here. Our main workflow is that we collect eggs from Devil's Hole. This is our other staff member, Olin Feuerbacher, who is our aquaculturist extraordinaire. We put these little egg mats out in the wild population on the shallow shelf area. They are really just carpet attached to a tile with little feet, so they don't actually sit on the substrate very much or have much of an impact to the natural habitat. We put these out once a month. They stay out for about 4 days. We bring them into the lab and then look for eggs. So any eggs that are found, we rear to maturity in the lab. We work with them to try to see if we can get them to spawn in aquaria, and then we stock them into the refuge tank. And here you can see just a couple of an embryo and a larval pupfish. So finding the eggs is a fun endeavor that we do once a month. And I'll give you just a couple of seconds to see if you can find the egg. Anybody spot it real quick? In the interest of time, I will show you where it is. But these little eggs are very precious, and our entire population has been started with egg collections. An adult pupfish has not been removed from Devil's Hole since 1998. So everything that I'm going to show you and tell you about today and all of those fish that you saw in that video came from eggs from Devil's Hole. So we rear them in our propagation room, which sounds a lot fancier than it looks like here. The Devil's Hole pupfish aquaculture methods were worked out by Olin Feuerbacher over the years at the facility here. And we raise them up to the size that we can take a fin clip so that we have a genetic sample of every fish that comes through the propagation room, and then they go out to the refuge tank. So over time, since the facility started functioning in 2013, just some data in terms of what we've done. Over 400 eggs have been collected. The number of eggs incubated, that is those that look like they're viable or fertilized. And we've produced over 180 adult fish over the years. So there's some variation across years that mostly reflects changes in our permit abilities. So when they first started, they were only allowed to collect eggs like 2 months out of the year. And now we're allowed to collect every month out of the year. 2020 has a little bit lower numbers as well compared to some of the others, just because we haven't been able to collect as often due to COVID. We can also just sort of learn a little bit about the fish from these collections themselves. They have 2 breeding seasons, so most of their breeding is early spring, and there's a secondary breeding season late in the year, peaking in November. So they only live about a year, and so this November peak is likely very young fish that are basically the ones that were spawned in early January and February, or February and March. And then because of that energy limitation, the breeding sort of drops off and then picks up again once you start to see sunshine hitting the shallow shelf again, and sort of driving the system. So we've had good success in these first 2 steps, but this 3rd step just has never really been successful. So all of the efforts to breed fish in an aquarium, in a lab-like facility, just have never worked. And this was really pretty well known for a long time. So this is a note from the 1969 Desert Fishes Council meeting, their proceedings, that says there, with the exception of Cyprinodon diabolus, which is devil's hole pupfish, no difficulty in breeding any species of Cyprinodon has ever been experienced in aquarium culture. So get yourself some pupfishes and you'll be good to go. Just don't get these ones because these ones don't seem to breed in captivity. So again, setting the stage for our work. These are the previous captive breeding attempts early on in that time period when they were first being protected legally. The Steinhardt Aquarium, which is associated with the Cal Academy of Science now, and then some researchers at Fresno State had some attempts, and then there were some researchers at the University of Nevada, Las Vegas that also attempted some breeding. All of them, well, from what we know from notes that we can find, all of them did produce some eggs, but most of those eggs were not viable. So if there's two numbers under viability, and you'll see this set up on a couple of slides coming up here, the first number is just all of the eggs that look good. That is, they're nice and clear, they look viable, they're not fungused over, they're not dead eggs. The second number is confirmed fertilized. So we know that they're viable. They either have eyes or a heartbeat, depending on who it is that's using that. But those are similar stages. So we've had some success. They had some success. Hatching rate, though, was very, very low. And then again, percent survival to adulthood was also low. This 66% and even 50%, it doesn't look that bad. That's a pretty good survival rate. Unfortunately, the 50%, that was 1 out of 2. And then the 66, that was 2 out of 3. So even though that those survived to adulthood, there was no second generation coming after them, especially from this one that it was like one sad fish that was all by himself. So I want to go back to the beetles. And so the beetle story became really, really important for us because it represented a breakthrough in our captive breeding efforts. So this, once it was identified that beetles were preying upon larval pupfish and eggs as well, we started collecting those in the refuge tank. And this piece of the puzzle, that we were never able to spawn fish derived from devil's hole in aquarium sorts of environments, we had another opportunity to try again with fish from the refuge tank. So by controlling the beetle population, that predation pressure, that allowed us to collect eggs from the refuge tank. And it really was night and day. They started collecting beetles in March of 2018. And up until that point had gotten, I think, 12 eggs total out of the refuge tank in years worth of efforts. They collected about 450 beetles in 3 hours, the first beetle collection. And then on the next egg collection from the refuge tank, they got 40 eggs. So it really seemed like controlling that beetle population is key to successful reproduction in the refuge tank. So now we have a second source of eggs, and we can try the same workflow with this group of eggs. And in this case, it worked. So we've been able to spawn these fish that are derived not from the wild population, but from the refuge tank in our captive, in our lab. So there's some really interesting ideas about this that we're hoping to look into in the future. And that includes some epigenetic work to try to understand why it seems like one generation removed from the wild, you'll start to get successful reproduction in a different type of environment. So this is our breeding setup. The first group of fish that we had success with in breeding were in this large tank on the left. Now we are actually able to successfully spawn fish in these 20-gallon aquariums over here on the right. And that has been fairly successful. So comparatively, With our breeding efforts, we've got Devil's Hole here on the top. Again, total eggs, percent viable, either looking good, or a second number would be like known fertilized, percent hatching, percent to survival, survival to adulthood, and then total adults produced. So we've had 2 breeding events in the refuge tank. One of them was in the summer of 2018 that produced over 1,000 eggs, but only 13 adults. This was largely due to needing to figure out aquaculture techniques that were successful to raise these fish. And then they started breeding again in the refuge tank last November. And we have collected eggs every month since then. So we've just last week completed a full year of breeding in the refuge tank with successful breeding every year. These numbers are a little bit lower in terms of total eggs. Because we were not able to collect as often due to COVID, and also because we reached capacity, having produced over 200 fish, we only have room for so many fish in our propagation room. And so we had to shut down our egg collections from the refuge tank in February because we were at capacity. We have been working with these fish though and breeding them in the propagation room. Again, here showing number of eggs produced and then our viability rates. And really the thing is that with each time that we get another chance, we seem to be able to improve. So we currently have a couple of groups of fish breeding in the propagation room and have produced over 50 individuals from these breeding efforts. So these are ongoing. We're still working out some of the aquaculture techniques in terms of just keeping these fish. Nobody's ever had 200 Devil's Hole pupfish to try to figure out what density they should be kept at, how often they should be fed. Some of these basic things we still don't necessarily know about the species. But it's a pretty exciting time because it gives us some opportunities. So another aspect that we're working with is another issue that was shown in previous refuge attempts was that the fish didn't really look like Devil's Hole pupfish anymore. And so again, one of these old notes. I love old notes. All the best ideas are like in old notes and really old books that people don't read anymore. So that's another just a weird piece of advice for grad students. Read all the old stuff. People have forgotten what they didn't know from there. But some of these refuge attempts were actually abandoned because of some changes in shape. So this is describing one at spring on Ash Meadows called Purgatory Spring. And what they went and found was that most of the fish were definitely larger than the maximum natural size for the species. They were misshapen with abnormally large abdomens. That's a nice insult right there. Misshapen with abnormally large abdomens. They actually proposed to no longer work with that population, and they did kill the population. So they just stopped the attempt. And this happens in all of the refuges. So this is a PCA type graph showing some geometric morphometric data. This is a representation of shape of Devil's Hole pupfish. The black squares are fish from Devil's Hole. And then the other two, the white triangle or the white diamonds and the white circles are Point of Rocks and School Springs refuge fish. So what we see is that they do change shape when they are in these refuges. And so this gives us an opportunity to evaluate what's happening in our refuge. Are we also getting these shape differences in the refuge tank in the fish conservation facility? This morphological question has been examined in depth by Sean Lima, who's at Cal Poly. And he was interested in the mechanism behind these shape differences. So compared to the other species of pupfishes, Devil's Hole pupfish are smaller, they're not as sexually dimorphic, and they are actually missing a set of fins. So they don't have pelvic fins. And what Sean did was he took a surrogate species and treated them like Devil's Hole pupfish. He limited their food because of that energy limitation. He jacked up the temperature and eventually did some manipulative experiments looking at thyroid hormone functions. And what he found was that if you take another species of pupfish that is usually much larger and has pelvic fins, and you don't give them much food, the percentage of fish with pelvic fins dramatically drops. And the same thing with temperature, and that does interact with food quite a bit. So if you treat other pupfishes like devil's hole pupfish, they will look like devil's hole pupfish. And it does seem to be mediated by a thyroid hormone mechanism. So this really is getting to understanding the phenotypic plasticity and the ecophysiology of the species, and understanding how their environment and the features of their environment have led to changes in the species. And so this mechanism is pretty well established. The water temperature influences metabolic rate, which combined with high or low food is really going to change the growth rate. The growth rate is going to interact with your thyroid hormone physiology, and then you either can get normal morphological development or neotenous morphological development, basically devil's hole pupfish style development. So one of the cool things about being able to produce devil's hole pupfish in larger numbers than ever before is that we actually have fish to work with for the first time. But the limitations of the Endangered Species Act and our permits do limit what we can do. So one of the things at the fish facility, in addition to just housing the devil's hole pupfish backup population, is that it is envisioned as a research station. And so we're starting to try to figure out what that looks like now that we are starting to be able to have fish that people could do research on. Much of what we've started to do is non-invasive. And this is where, tying back to Sean Lima's work, this really comes in, because we can look at growth rates by taking pictures and not having to do anything that would potentially harm or stress the fish. So we took some of his data and looked at his early stage growth rate data. So it's a standard length, and he is looking at days post-fertilization on these different food treatments. And we can take this data, or this information, and make some predictions. So we expect to see our fish reach about, we'll say 10 millimeters at different days post-fertilization, depending on, the kind of food regime. We give them probably a medium food regime because we know that we don't want to overfeed them. We also want to give enough foods so they're not on a super low food regime. So this gives us something to compare to. And we've started to do this. So this is a just hatched devil's hole pupfish, and we're tracking growth rate at post-hatch of all of our different groups of fish. So our predictions on high food are that we'll reach 5 millimeters by about 5 days post-hatch, and 10 millimeters at about 25 or 30 days post-hatch. On a low food, it gets extended quite a bit. The light blue bars here are eggs derived from devil's hole, so larval fish derived from the wild population. The dark blue bars are fish derived from our captive population. So we can use this information, start to understand how phenotypic plasticity is working in actual devil's hole pupfish, not just a surrogate species, as well as evaluate differences between wild and captive derived fish to see if there are any differences in our captive population. So we've got a lot of overlap sort of in the first week of life, and then we're still adding data to this with additional cohorts. But we start to see a little bit of variation, which I don't know if it's statistically significant or not, but by about 2 weeks old, it does seem like our refuge tank derived fish might be having a lower growth rate than the wild derived fish. So this is just sort of really preliminary data, but giving us sort of a direction of where to go, and hopefully illustrates, some of the science and some of the questions that we're starting to look at in our facility, in addition to contributing to recovery of the species. So one of the keys really was that recovery of the species depended on understanding community ecology. And with our beetle trapping efforts in the refuge tank, that really was sort of the key thing to allowing both the refuge tank population to thrive, as well as allowing us to build our captive population in our lab environment to have fish to work on. So we have a lot of different types of questions that we are going towards and working to. My personal philosophy is that conservation and management requires all different types of approaches and levels of analysis. And this is very much an animal behavior, Tinbergen's 4 questions kind of structure. And there has been research on all of these things in Devil's Hole pupfish. So some of the questions that people have looked at are trying to understand the evolutionary history of the species and gene flow between Devil's Hole pupfish and the other species of Cyprinodon that are found throughout the Death Valley Basin. We're continuing to look at this and are doing some genome work to try to inform this question, as well as our management-based questions. Understanding the physiology, in addition to the work that I just told you about, looking at the endocrine and the thyroid hormone mechanisms, there might be some other hormone mechanisms that are important to understanding how they work to maximize our breeding capacity. They also do some really strange things in terms of their metabolism. We know that they can go for hours without using any oxygen. And that's another really interesting thing, especially in the context of climate change, to understand for vertebrates. The adaptive value, this is sort of your functional questions. These are your sort of classic behavior questions. And there's no sort of published paper things on here quite yet, but that's mainly because sample sizes for behavioral work tend to be quite a bit bigger, and they haven't existed in the past. So we have a lot of questions about foraging behavior with their energy limitation. There have been questions about personality and behavioral syndromes in other species of pupfish and how that might influence conservation efforts, as well as just understanding mate choice and sort of classic sexual selection questions in the species. Those are really key to getting something to breed. And then the last one, ontogeny. We are tracking all of our eggs that come through the facility now, and we've seen some very interesting differences in those developmental stages compared to a closely related species where we do have a developmental staging series published. So we'll be looking at development very strictly in terms of larval development, but also development of breeding behaviors is another one that's really important, because we've discovered or just found that they breed at a much smaller size and a much younger age than anybody had thought. So by being able to have them in a lab and observe them on a daily basis with a lot more attention, we're starting to understand a little bit more about the breeding ecology as well. So with that, I think I have time for questions even, if anybody has any.
Yeah, a few minutes for questions, folks. If anyone has any, jump in.
Hey, Jenny.
Good.
Hi, I'm Leon. That was a really great talk. Thank you for that. So I'm just curious. This seems to be a very extreme environment in terms of like temperature and oxygen, like you were talking about. So I'm just wondering, do you have historical environmental data that goes back to like the 70s when this work was starting? And do you see like a lot of fluctuations in temperature and oxygen?
Right. So I'm not sure how far the historical records go back in terms of tracking that. I'm sure that they go back at least into the 70s and 60s when people had done some collections and things like that. From what I understand, the temperature is fairly stable because it's geothermally heated. So the interesting thing is that that's the temperature of sort of the deeper areas of the pool or of the cave. And so because of the volume of water that's actually over the shallow shelf is so small and the depth is so small that that you get a lot of variation in temperature on that shallow shelf. So when it is hot in the summer, we get into the 120s pretty regularly. You can actually get that shallow shelf area topping out at 37, 40 degrees C. So just because that areas, so shallow, the volume is smaller, so you get a lot more temperature variation there, and dissolved oxygen variation as well, which is tied to the nice Spirogyra algae that you get there. So we think that there might be some preferential egg laying in relationship to the algae that grows on the shallow shelf, but haven't done anything to test that. But that would definitely be tied to the variation in dissolved oxygen for the eggs.
Thank you.
Hi, this is Spencer Lashia. Thank you for your talk. It was incredible. I was wondering if you could elaborate a little bit more on kind of how the I guess the forecast of the species kind of influences the questions that you asked. You showed a graph just looking at the probability of this devil's hole pupfish's like probability of extinction over 50 years, and I was wondering if you could just elaborate a little bit on what kind of questions that you guys asked, particularly because of that kind of forecast.
Right. So that's a really interesting question. And I think that I'm not sure that the team has really fully embraced that. That was published in 2014. And so we're, 6-7 years into that 50-year forecast already. But the team as a whole has worked on a strategic planning document that really identifies triggers of all sorts and responses. And it's interesting because in that planning document, we tried to sort of look at all possible things that could happen, but we didn't really go all that far into the extremes. So that's still on the table for us to do, is to think about, really long term. I think the planning document that we're working on it's more of like a 10-15 year kind of a thing instead of a 20, 30, 50 kind of year thing. Because that's one thing. So we do not move fish back out into the wild. And that's definitely something that would need to be considered in that case, and it would depend on the reason for why we think it went extinct. So there's just demographic stochasticity, which plays a role, but there are also concerns about the genetic variation in the species. People have, over the past 20 years or so, sometimes gotten into really spirited arguments about whether or not this is like a mutational meltdown situation, and lots of fights about genetic rescue and introducing another species into this species and sort of trying to do some directed hybridization sorts of things to try to increase the genetic quality of the species as a whole. Kind of like a Florida panther thing that they did, but those were the same species, just different populations. So I think that it's something that we need to think about more, if it's the genetic situation that is causing issues and we're seeing declines in the population, that's a different fix or a different approach required than if it's climate change. And so I think we're getting there, but I think we'll need to be thinking more about that and some of those extremes. But we try to keep things in mind and take all different kinds of approaches. The genetic one is very high on our list of things that we're looking at.
Thank you very much.
Okay, so we're just about out of time. Maybe if there's one more short question.
I have a question. It's sort of a follow-up to that question. So I thought it was really interesting where you showed that you guys were able to recreate the pelvisless phenotype in the surrogate study species.
Mm-hmm.
And, so it got me thinking about this one paper I think I read where someone had thought or proposed that this pupfish was introduced some hundreds of years ago by Native Americans.
Yep.
Right. So I'm just wondering, and I also had some other questions for you about your captivity thing, but I've never actually talked to anyone that's worked with these pupfish. So I just wondering what the pupfish community thinks about that 200-year ago introduction idea, seeing as
Right.
the type evolved so rapidly in the lab.
Yeah, so the that is this series of papers back and forth. There's like two groups that have done work that have estimated the age of the species, and I think the most important thing to keep in mind is that age of the species just means the last gene flow. It's the last time that anything else got introduced or in any way to Devil's Hole. So the estimates do range from 180 years to 60,000. And it really depends on your parameters. And ultimately, all of the age estimates depend on what you use for your mutation rate. And there is other evidence in other species of pupfishes that do suggest that they have some of the highest mutation rates of any vertebrates that we know. So so those things are things that we would need to know in order to really get an idea. Ultimately, they are morphologically different. They are evolutionarily different. And so, however you want to define your species, they're still worth conservation time and effort and resources. Also, the species probably wouldn't be in such a perilous situation if it wasn't for the actions of humans and pumping the water. So to a certain extent, the group that works really directly on the conservation and recovery of the species, like we don't really care about the age because it doesn't change anything that we do from a scientific standpoint. And just a pupfish researcher, I think it's fascinating. I think we'll get to some of those questions. We have a contract sort of in place with a collaborator to do a de novo genome assembly of Diabolus and then one of the closely related species. They're actually looking at historic losses of variation and have some really, really exciting data finding deleterious mutations over time from historic samples and museum samples to current. So I think some of those questions will be answered by some of the work that we're doing along those genome lines. But in terms of the day-to-day operations, like it doesn't change anything about what or how we do what we do. Hopefully that answers your question.
Yeah, thanks. Thank you.
All right, so we should probably end things there, but thank you very much, Jenny, for that talk. Devil's Hole pupfishes are just amazing. So thanks again.
Yeah, thanks for the invitation and thank you very much for all your attention. I appreciate it.